Hello, welcome back to my channel by Shansi. In today's lesson, I want us to look at how to use a calorimeter. You will be asked how a calorimeter is used. You'll find this in your exam papers where they are asking you the advantages of using a calorimeter to track or to measure the progress of an enzyme controlled reaction. And why do we need a calorimeter? You can see this word color. The word calorimeter has the word color in it. So a calorimeter is mainly used in reactions where we have color changes as the reaction occurs. Like an example, imagine we are having a reaction where we have amylase breaking down starch to form a reducing sugar. And maybe by the end of the reaction, you are looking out for the presence of reducing sugars. So you might see color changes, you might see blue, you might see green. Yes, we can interpret these color changes because we know blue means reducing sugars. Green means reducing sugars present, but they are little. And then brick red means we already have a lot of reducing sugars. But these colors do not exactly give us an idea of how much of the reducing sugar is there. These colors are just qualitative. Qualitative, we don't know how much, what is the concentration of the reducing sugar in each test tube. And that is how now a calorimeter comes into play. So if the method used for measuring the progress of an enzyme-controlled reaction involves a color change, you've had examples of different experiments where you expect a color change to occur. Maybe the test for proteins, I'd expect purple to show presence of the proteins. So whenever we have a color change, then that is when a calorimeter comes into play. How? The calorimeter is able to get that color change and measure it quantitatively. In other words, it's able to measure how much of the substance causing the color is there in a solution. Yes, that is how a calorimeter is going to be used. And let's look at how a calorimeter works. A calorimeter is very simple. You're going to get your colored substance. It uses small tubes. So those small tubes are called cuvettes. This is the spelling of the word cuvette, C-U-V-E-T-T. So they are small tubes. So just get your solution that has color or your colored solution, and then you place it into a cuvette. Now that cuvette is going to be inserted into the machine, which is the calorimeter. The calorimeter has lights. It has a bulb that produces light. So when the bulb produces light, that light is made to pass through the solution. The light is made to pass through the solution. And as the light passes through the solution, now because this solution is colored, remember, colored substances usually absorb light. So because this solution is colored, it's going to absorb some of that light. And depending on how much light has been absorbed, the calorimeter is going to show you a reading. The reading is what we call absorbance. The reading is called absorbance. So it shows you absorbance of light. In other words, if the colored substance is a lot, we shall expect a lot of absorbance. So the absorbance depends on how much of the substance causing the color is in the solution. If the substance causing the color is a lot, it means the solution will be so much colored with a high intensity, and still that means that the absorbance is going to be high. If the substance causing the color is low, then it means less light will be absorbed. And then at the same time, the absorbance is going to be low. So this value here is a numerical value. You can see now we have figures. Instead of us just having blue, red, brick red, orange, purple, we are having figures which can even be plotted on a graph. Remember, on a graph, you cannot just plot color on a graph, but at least the absorbance can be plotted on a graph. So a calorimeter provides you with what we call absorbance. So with absorbance, we look at how much light has the solution absorbed. And in some cases, you even find there a filter. I had not talked about this apparatus here, the filter. Why do we place there a filter? Remember, different colors absorb different wavelengths of light, or different colored substances absorb different wavelengths of light. So if your solution is blue, you might need to know what is the best wavelength of light that blue color absorbs, such that you provide it with the best wavelength of light. That is why we have this filter in place. So the filter is to allow the right wavelength of light that can be maximally absorbed by the solution to be exposed to that solution, leaving out other types of light or other wavelengths of light that might not be absorbed, so that we have the best absorbance recorded. So that is how a calorimeter works. Now, after you have gotten your absorbance, you record it down. So you might have another solution whose absorbance you still need to obtain. And that takes us to what we call calibration of the calorimeter. So I have this term, calibration. Calibration is more like returning the calorimeter back to zero. Calibration. So by the time you remove this solution, it would have given you the absorbance, but then you want to measure the absorbance of another solution. So it will not turn itself back to zero. You have to do that. And how are you going to do it? Usually when we are calibrating, we can get a colorless solution, which will give us an absorbance of zero. And in that case, the example of the colorless solution we can use is water. So get water, place it into the calorimeter, make light pass through. Because water is colorless, it will not absorb any of that light, and it will give you a reading of 0, 0. Then you would have calibrated your calorimeter ready to measure the absorbance of the next colored solution. Let's take an example of a reaction where absorbance needs to be measured. Assuming we are looking at a reaction where I have this enzyme amylase breaking down starch. So I could have starch, and then the starch is being broken down by amylase. To reducing sugars. In that case, we have the reactants and the products. The products are reducing sugars, which is maltose. The reactant is starch. The enzyme is amylase. Now, some reactions can be tracked using both the product and the reactant, but usually you just have to choose one to use. Like in this case, you are having starch being broken down to reducing sugars, and someone was tracking, investigating the progress of that reaction, finding out how fast the amylase is working. So we can be able to test for starch. Remember, to test for starch, you simply use iodine solution. To test for reducing sugars, yes, you use Benedict's solution. But according to this picture, you can see that they tested for the presence of starch. So as the reaction goes on, test for the presence of starch at different time intervals. An example, you could find that maybe they tested for the presence of starch at 0 minutes, then maybe at 5 minutes, then maybe the next is 10 minutes, 15 minutes, and then 20 minutes. So they could have a sample where we have the starch. So a sample where we have the starch and the amylase. mixed and the reaction is going on. But at different time intervals, they could pick off a sample from the reaction mixture and test for the presence of starch. Now you can see in this case, we are just having color changes. When you look at the color at 0 minutes, it's almost similar to that at 5 minutes. And someone might think, the starch is not being broken down. However, the reaction is happening, but our eyes are subjective. Our eyes might not be able to tell the color differences. That is how a calorimeter really comes into play. Yes. So the calorimeter will be able to even detect the smallest changes or the smallest differences in concentration of the substance causing the color. You'll find that the amount of starch in test tube 1 is not the same as the one in test tube 2. But if you just use your eyes cannot be able to identify that. However, if you use a calorimeter will be able to identify the difference. How? The absorbance in test tube 1 that it will give you will not obviously be the one as in test tube 2. Yes, for the test tube after 20 minutes, it's very much obvious that yes, the levels of starch have really so much decreased, but still its absorbance can be measured. And if we measure absorbance, we can see that obviously test tube 0 will give us the highest absorbance. Why? Because the color intensity is a lot, meaning the substance causing the color, which is starch, is much. Hope you remember how to test for starch. You just use iodine solution. You get your starch solution, add iodine solution, and then observe the color change. So in this case, we are having these different samples, but you can see they are all colored. So we can measure the absorbance of each of these test tubes. And the results, we can predict the results. Test tube 1 will obviously have the highest absorbance, and last test tube, which is test tube number 5, will have the lowest absorbance. So that is how a calorimeter works. It allows you to track the progress of a reaction, especially a reaction that involves color changes. A reaction that involves color changes. So you'll be using a calorimeter. Yeah, we have already talked about this photograph. It shows you a range of colors obtained with the iodine test during the course of an experiment where they were investigating the digestion of starch by amylase. So in that case, these test tubes show us increasing time for digestion. Obviously, the one at 20 minutes, much more time was provided for the enzyme to react with the substrate. That is why most of the starch had been broken down compared to the one in the beginning. I've talked about this already. So in that amylase experiment, which I have just described, the intensity of the blue-black color can be measured using a calorimeter, and the color is acting as a measure of how much starch is still remaining. So the higher the absorbance, it means the more starch. The lower the absorbance, it means the less starch. And in that case, we can even plot a graph. If you go back slightly here, remember we were having time. We had 0 minutes, we had 5 minutes, we had 10 minutes, we had 15 minutes, we had 20 minutes. So in this case, by the end of the investigation, would have obtained absorbance for all these 5 test tubes. And then we can even plot a graph of absorbance against time. Those are some of the advantages of a calorimeter. It gives you quantitative results here. It gives you numerical values, which you can be able to plot on a graph. Unlike if you just had blue-black, light blue, brown, that wouldn't be possible. So if you do this over a period of time, you can plot a curve of amount of starch remaining against time. Amount of starch you can see is in quotes, which will simply be the absorbance. And then you can even be able to get the initial rate of reaction. And here they are telling us, since they were using iodine, you can even be able to mix the starch and iodine, and you look at how the colors change as the reaction goes on because you have mixed them at the same time, starch, amylase, and the iodine. However, sometimes these chemicals can interact with the enzyme and prevent the enzyme from working at its best. That is why it is better you do not get the iodine and you mix it with the reaction mixture. Just get off a sample from the reaction mixture and then add the iodine and see how much starch is present at that particular time. So advantage of using a calorimeter, number one, it provides you with numerical values, figures, which is the absorbance. And then those figures can even be used to plot a graph. That is another advantage. The readings are very accurate. It is able to distinguish small differences in the intensity of color. So those are some of the advantages of using a calorimeter. Please, if you feel this video has helped you, give it a thumbs up. Don't forget to subscribe to our channel. I'm always going to be uploading videos, uploading revision material. Kindly subscribe and share this video with your friends so that you do not pass alone. See you on the next one. Bye.